Mississippi. After completing her Bachelor of Science in Biology, Caitlin went on to complete a PhD at Texas A&M University, where she utilized various molecular techniques and next-generation sequencing technologies to study the skin microbiome and cytokine profile in feline allergic dermatitis. She's now using her molecular biology and bioinformatics experience to study microbial species relevant to catfish health and production. So, Caitlin, take it away.
Thank you for the introduction and the opportunity to present today. Today, I'm going to present about nanopore sequencing for aquaculture bacterial microbiota profiling. Before I get to the actual presentation, I do have to put up this disclosure, just because I'm mentioning quite a few trade names, and this is just for the purpose of providing information, not any kind of recommendation or endorsement by USDA. So we all know that bacteria play integral roles in fish health and production. And so profiling these communities can be really important and can be useful for many different purposes, including identifying dysbiosis associated with disease outbreaks, evaluating the effects of management practices, for example, using antibiotics, and understanding the influence of alternative ingredients. The concept of profiling bacterial communities is not novel to the last few decades or anything like that. It's been done for years and years. However, the way that we have done that has changed. Previously, this was mostly done with bacterial culture, but as I'm sure most of you know, bacterial culture is not always easy. Some bacteria just don't grow as well in the lab, and it's not always, you're not always able to distinguish between two bacteria on culture. And so sequencing-based approaches have really taken first place as a method. And in particular, high throughput sequencing of the 16S rRNA gene has become the predominant method used. It allows for broad characterization of the bacterial microbiota. So I'm guessing most of you already know how Sanger sequencing works, but I think it's helpful to compare Sanger sequencing to high throughput sequencing for this presentation. So usually with high throughput sequencing or with Sanger sequencing, you might perform a PCR and generate a clean PCR product, which you then subject to synthesis reaction using these fluorescent nucleotides, which are then size separated on gel and read through a machine that gives you your sequence. However, in a complex sample, like a fecal sample where you have multiple bacteria, if you run a 16S PCR, you're going to end up with multiple DNA fragments that are different. So it's going to look something more like this, where you have these 3 DNA amplicons that are of the same size, and it's really the same gene, but they have different sequences. And in the end product, your sequencing results are going to look more complicated. So you're going to have all of these ends as bases instead of actual called bases, because you have different nucleotides at each base. So you'll end up with a poor quality sequence, which prevents you from identifying the bacteria that are present in your sample. With high throughput sequencing, you can use these 3 different sequences or amplicons that you have in your PCR product, subject it to the magic of sequencing, and then you end up with all of your sequences produced as distinct reads. So you're able to identify every single bacterial species that's present in your sample. And so this is really useful for profiling bacterial communities that are in complex mixed samples. There's quite a few different platforms that can be used for 16S profiling, but I'm just going to focus on 2. The first of which is Illumina sequencing, which is similar to Sanger sequencing in that it uses sequencing by synthesis, but this is a high throughput sequencing platform. And this technology specifically produces really high quality short reads. So with the machine that's shown here, this is the Illumina MiSeq, you can generate reads of up to 300 bases. But if you do paired end sequencing, meaning sequencing in the forward and the reverse direction, and then you merge those reads, you can then have up to around 500 bases of a sequence. So this can allow you to sequence up to 1 or 2 informative regions of the 16S rRNA gene. Oxford Nanopore is a newer sequencing platform that hasn't really been adopted widely by the microbiome field. And this is a different technology entirely. So this uses single molecule sequencing, where there are bacterial pores where a single DNA molecule goes through and is sequenced. And so Due to the nature of this technology, you have a relatively lower sequencing quality, but you also have longer reads. Some have reported getting reads up to 2.3 megabases. And so clearly for the 16S rRNA gene, that means we can sequence the entire length. And so this seems really attractive because if you're able to sequence the entire length of the 16S rRNA gene, that's more room that you have to distinguish between individual taxa. So you can potentially have higher taxonomic resolution, potentially be able to tell different species apart rather than just different families or different genera. So I really wanted to evaluate if this Oxford nanopore sequencing would be useful for aquaculture research. So to do this, I had 3 aims. The first was to compare the accuracy of nanopore and Illumina sequencing of an aquaculture relevant mock community. Second was to evaluate the ability of these protocols to identify differences between 2 experimental groups. And then finally, to compare the results of these methods from several different aquaculture relevant sample types. So first I needed to produce a mock community. So there are some mock communities that are commercially available, but typically these contain bacteria that have more relevance to human health. So what we did is we took 11 different bacteria that are relevant to fish, or at least found in fish, grew them up in culture, transferred them to broth culture, pelleted, and then extracted the DNA. We confirmed the identity of these species either with Sanger sequencing or qPCR, and then combined equal proportions of the DNA at the genus level. So although we have 13 bacterial species, these are incorporated as 8 different genera. All of the data that I'll show from here on out with the relative abundance of this mock community is after correcting the proportions based on the number of 16S copies and genome size for each species. We also wanted to get realistic samples. And so we had 1 pond with hybrid catfish. And from this 1 pond, we took 5 water samples. We also took 10 hybrid catfish, put them into 1 holding tank where they were held during sample collection. From this holding tank, we took 3 water samples. And then of these 10 catfish, we split them into 2 experimental groups. The first group were fish that were euthanized with an overdose of MS-222. And then the second group was euthanized with cranial concussion, followed by pithing. From these fish, we collected digesta and then also swabbed the gill and skin with swabs and extracted the DNA. After DNA extraction, we subjected the samples to nanopore sequencing on a gridiron with 2 approaches. So the first one I'm calling the ONT16S. This uses their commercially available kit for 16S barcoding, which targets near full length 16S sequence using the 27F and 1492R primer. And then we also use this protocol that I'm calling the ONT custom, which does use a commercially available kit, but with custom primers. And the difference between these primers is just the forward primer has a few more degenerate bases, which should allow more wiggle room for more bacteria to be targeted. The data that results from this were filtered with nanofilts, and then I got taxonomic assignments by using Minimap2 and the Silva 138.1 release. The DNA was also used for Illumina sequencing on an Illumina MySeq using 2x300 paired end sequencing chemistry on amplicons of the V4 region of the 16S rRNA gene using the primers 515F and 806R. The resulting data were first pre-processed in QIIME2, and then the data from both the nanopore and Illumina sequencing were then imported into R, where all of the diversity analyses and statistics were performed. So starting off with this first aim, here we're looking at the relative abundance of the genera in the mock community in a heat map. So here we have the theoretical relative abundance in our mock community, and then we can see that our ONT custom data was the most similar to our theoretical, followed by the Illumina and the ONT16S. And this was also supported by the root mean square error values, where ONT custom had the lowest value. An important finding was that Yersinia was not classified to the genus level in Illumina data, but it was present at an appropriate relative abundance at the family level. So this reflected an issue with classification, or perhaps with this region not covering a part of the sequence that is different enough between different bacteria of the Yersineaceae family. Bifidobacterium was the bacteria with the most variable relative abundance. You can see that here, so it looks pretty similar between the theoretical and the ONT custom. It should have been present at about 19.5% relative abundance, but in the ONT 16S, it was present at 5.7%, and in the Illumina, 31.3%. So you'll notice here that we analyze this data at the genus level, but I said that we included different species. Illumina data typically doesn't allow you to get to species level identity, and so we didn't analyze the Illumina data at species level, but we did look at the nanopore species level. So here we're looking at the different species that are present. Here is the theoretical composition of the mock, and then our ONT custom and our ONT 16S results. Due to the way that these kits are made, we can only multiplex so many samples on a single flow cell. And so we had to split our samples across 5 runs for each of these protocols. So the purpose of this was really to look at how consistent it is across runs. And it does look fairly consistent across the different runs. One thing that you might notice is a lack of this purple, which is Francisella salamarina in the actual reactions. And this bacteria was present at the genus level, so Francisella was present, but the specific species, salamarina, was not present. Instead, there was a high abundance of an unclassified species. It turns out that this species is actually not found in the Silva database, and really the Silva database isn't even curated at the species level. While the sequence is present in the database, the taxonomy is not. So I used an alternative taxonomy for the Silva database and was able to find that 99% of the sequences that corresponded to Francisella unclassified were actually Francisella salamarina. And so the graph really looks like this, and this just reflects an issue with the database itself. On to the second aim, where we were looking at 2 experimental groups, in our case, euthanasia method. Here we're looking at beta diversity. So what this looks at is how similar different samples are based on community structure. So after computing beta diversity, these are then plotted here on principal coordinate analysis plots. And each dot represents a single sample. Dots that are really close together are more similar to each other than dots that are far apart. Here we have the Illumina data, the ONT custom, and then the ONT 16S in the skin. And this is all with the Bray-Curtis method. However, this was also done with the Jacquard index as well. And there were significant differences found for skin samples in all of these different sequencing protocols, which was quite interesting. There were some significant differences with Gill and Digesta as well, but overall the skin was the most different. This was also found when we looked at differential relative abundance testing. So here we have names of bacterial taxa that are found in higher relative abundance. So the ones with the blue bars are found in higher relative abundance in the samples from fish euthanized with MS-222, and vice versa for these ones that are in peach. And so you can see there's quite a few taxa that are found with differential relative abundance in the skin. And there were some as well found in the gill and the digesta. These results were obtained when I did an analysis incorporating all of the data from the 3 different sequencing methods. However, when I also split it by sequencing method, there were significant results in all analyses of the skin. So really the skin has quite a few differences. And I think what was really interesting from this analysis was that there was a difference in the number of significantly differentially abundant taxa. So with the Illumina protocol, 55 taxa were identified as being differentially abundant. However, ONT 16S and custom only identified 36 and 25 taxa. So this indicates that the Illumina data is more sensitive to differences between euthanasia method. That does not necessarily indicate that the Illumina protocol is better for doing this, because we don't know the true composition of the samples. However, it's important to know that there are these differences with protocols. The last aim, we wanted to look at real samples that are of relevance to aquaculture research. So first, we're looking at the sequencing success, which is presented as the % of breeds that pass the quality filters and were classified as bacteria. The digesta and the gill swab both resulted in significant statistical tests. However, only the gill swab yielded significant post hoc tests, as you can see with these superscript letters. What you can see is that overall, the ONT protocols had lower sequencing success for gill samples, which was due to shorter DNA fragments. So with Illumina, we only need a 300 bases of sequencing, but for the Oxford nanopore sequencing, we needed at least 1,200. And so since there were so many short reads, a lot of them were discarded during the filtering process. Here we're looking at beta diversity again, but this time of all the different sample types and sequencing methods. So here we have our shapes corresponding to our different sample types, as you can see here, and then colors corresponding to sequencing method. And what's really obvious is that sample type is more impactful than sequencing method. However, sequencing method does have a significant influence on the skin, tank, and pond samples. So if you just look here at these crosses, which are the pond samples, you can see that the Illumina samples really cluster tightly and separately away from the nanopore samples. And that was the same as well for the tank and skin samples. Now we are looking at alpha diversity, which is within sample diversity. So you calculate the number of, here in this case, the number of genera or the richness or evenness in each sample. And what we could see is that alpha diversity rankings among sample types was consistent across the sequencing methods. However, the calculated diversity was different between methods. So what this means is if we look just at this first graph, which is Chow-1 index, the alpha diversity rankings were consistent across sample types. So regardless of sequencing method, skin was always most diverse, followed by tank, ponds, and digesta. However, if we focus just on skin samples, you can see that both the ONT-16S and ONT custom protocols identified higher diversity than the Chow-1 index. And this was after collapsing all the data just at the genus level. And finally, we're looking at the relative abundance of the different bacteria across all of the different sample types. So on the top, we have our Illumina, in the middle, our ONT-16S, and then our ONT custom at the bottom, and then our different sample types across in different columns. So each color represents a different phylum. So all of the orange is firmicutes and all of the purple is fusobacteria. And within each color, the different shades represent different genera. So if you just looked at the phylum level, it would look pretty consistent. Like if we're looking at digesta, the firmicutes are the most abundant in some of these samples, otherwise it's fusobacteria. But when you get to the genus level, you start to see some differences. So for example, here you see a lot more of this Rhodobacter than you do in the nanopore data, where Plesiomonas is the most predominant proteobacteria genus. So you can see that there are some consistencies, but really it's quite different between the nanopore and the Illumina. And I think this is really interesting because with our mock community, we saw that the ONT custom was the most accurate. However, and the Illumina was the second most accurate, but here the two nanopore protocols look pretty similar. So this is why it's important to test it not just in the mock community, but also in these experimental samples. So on to conclusions. The ONT custom protocol produced the most accurate mock community results, but as I just said, in real samples, the ONT custom and the ONT-16S looked pretty similar. The sequencing protocols differ in their capacity to identify differences between experimental groups. We saw that the Illumina identified way more differentially abundant taxa than either ONT protocol. I think it's really important to emphasize that your choice of sequencing method depends on quite a few things, like your sample type, the number of samples that you have, your access to equipment, and also your bioinformatics support. But overall, I think nanopore sequencing has been proven to be a viable alternative to Illumina for bacterial microbiota profiling of aquaculture relevant samples in certain situations. As for future directions, there's quite a few places I could take this technically. So I think there's a lot of room for improvement in the protocols, especially for the gill bacterial microbiota sequencing. Also, it would be nice to assess protocols that allow for an increased multiplexing ability for the nanopore sequencing, since currently we had to split across multiple flow cells. And obviously, it would be nice to look at databases that have informative species level identification that can correctly identify that Ference salamarina. But I think what's really more important than these technical future directions is actually applying these to problems of importance to catfish farming and farming of other fish species. And I'd like to acknowledge all of my co-authors on this study, as well as the University of Minnesota, who performed the sequencing, and the website where I got a lot of the icons. And I'm happy to take any questions.
Excellent. Thanks a lot, Caitlin. Very well done. As before, if anybody has any questions, please put them in the chat box, or go ahead and unmute yourself and ask Caitlin directly. We've got one here. Nice work. Which DNA extraction protocols did you use for the swab samples, and how was the DNA yield for those samples? You're muted, Caitlin.
Sorry,
There
great
goes your
question.
perfect day. Yeah.
So we use the QIAGEN Power Soil Pro DNA isolation kit, and it gave me a very false impression of the gill samples because I just had so much DNA, but obviously that doesn't just extract bacterial DNA, you have a bunch of other stuff. So the DNA yield was fairly good for all of the samples, but that didn't necessarily translate to after doing the 16S PCR and a bead cleanup.
The esteemed Dr. Esteban Soto has a question.
Hey, Caitlin, great presentation. And probably you guys have discussed this, as I know Matt has brought it, in previous conversations, but how do you account, for all three methods, you're basing yourself on a 16S? And particularly when you look at the Flavobacterium group that we know that have multiple copies of the 16S, how do you account for a single bacteria having multiple copies when you are estimating abundance in these populations? If a lot of the bacteria, you might not know their identity. So how do you account, for bacterium that have multiple copies of the 16S to assign them to a single species or a single organisms with either of these three different methods that you're investigating?
That's a good question. So unfortunately, that's not something that I believe has been well addressed by microbiome research in general. While we're all aware that there are differences in the number of 16S numbers, that's not something that's well incorporated into the analysis workflow. At least for our mock community, we can incorporate that, but since we don't know what we're getting in our communities, we can't really get it. And this is why it's really a relative abundance, and even then it's not a perfect relative abundance. It's not really showing everything, if that makes sense.
Hey, Caitlin, I suppose I could ask you this, not in front of everybody, but I'll just do it here anyway. Do you have any theories on why the euthanasia method would influence results differently based on the platform? It seems like if concussion and pithing is going to influence your results, it would influence the results equally and not differentially between the methods. So do you have any theories on that?
One of the important things to note is this isn't a exact comparison of Illumina and nanopore, because with that Illumina, we're targeting the V4 region, whereas with the nanopore, we're targeting the full length or near full length 16S. So it's already not like apples to apples, So I'm not really sure why. I mean, the two nanopore protocols look pretty similar, so I'm guessing it has a lot to do with the region that we're targeting, but I'm not sure. I mean, I have a lot of questions about the euthanasia method comparison that would be interesting to investigate, but are not at the top of my list. I think the important thing from this is just seeing that there are differences. So when you're trying to compare your results to someone else's results and they're using a different protocol, it's important to keep that in mind, that maybe that's why you don't see the same kinds of differences, And also to go with one method when you're doing a study, don't choose don't go half the fish are just with MS-222 because it's convenient, and the other because you ran out of MS-222, you're doing cranial concussion.
I don't know who would do something like that.
I don't know if you run out of it. You didn't plan it well enough, start over.
That's one other, I don't know if anybody else got one, but while I got you on the line. So this gives you a little bit more freedom in the length that you can do, which gives you a little bit more resolution, but obviously you're now sacrificing that a little bit for the accuracy. Do you think this could, in theory, open up avenues to other genes that may be a little more informative and a little more divergent than, say, 16S, and maybe better equipped to ask or answer Esteban's question or address his concern?
Yeah, potentially. I know there was one study where they did, it was like some almost 5 kb fragment, and they were comparing it to 16S, but I honestly can't remember because it's been a while since I read it, what they had come up with. I can't remember if they were doing like the whole RRN sequence or something, but I think that could be really useful, is now that we have this ability to sequence a larger length, and maybe we go with something else so we're not dealing with the 16S issues. And not to mention that the 16S isn't perfect for species level identification. Some species have near identical full length 16S sequences, and alternative regions may be, or alternative genes may be better for resolving those species.
Yeah, I agree. Tom Locke would agree too, if he's still on the call. If anybody else has any questions for Caitlin? Going once. All right. Thank you all. Thanks to both of our presenters.